Bismillahirrahmanirrahim. 9700 November 24 question paper 5-2 question 2 explained in this video. Question number 2 was of course also for 15 marks. Question 1 was for 15 marks and this was for 15. The two-spot ladybird Adalia bipunctata is a species of small flying beetle that is found in northern Europe and other parts of the world. The wings of these beetles are covered by two tough structures known as elytra, and this is what they're showing you, the protective elytra. A bipunctate is an important predator of insect pests such as aphids. So this bipunctata eats up these aphids. These insects must feed on plants, including many crop species. Population numbers of A. bipunctata fluctuate in response to changes in the population numbers of their prey. And what are they preying on? They're preying on the aphids. The elytra of A. punctata shows phenotypic variation. Phenotypic means physical characteristics. These insect pests, here I'm reading here. The three most common morph or the forms found in northern Europe are known as T. typica, Quadrimaculata Q, and Sexpustulata, which is S. So three types, T, Q, and S. Forget what's the difficult names they're given to it. The typica, which is the T morph shown in figure 2.1, here is shown here. The T morph is mostly red with two black spots and is described as non-melanic. So this one is non-melanic. This is non-melanic. The other two morphs are melanic. So these two are melanic and this is non-melanic, but it's written here as well, non-melanic. So the typical morph mostly is described as non-melanic. This is the one which is red in color and with the two black spots. I'm sure you've seen them and I remember seeing them. The other two morphs are melanic and they're mainly black with some red. The distribution of the colors in these three morphs is shown in figure 2.2. So we can see this is the red part and then this is the black part. So the red part, let me shade it red. So this is all red, right? Here we've got only this place red, just the shoulder pads are sort of red, and here we've got this part red only. And then we've got these dots red as well. So this is red as well. Now I've done the drawing for you. And this is all red. I haven't shaded it all red, but this is all red. This is all red. Very nice. Now let's read what they're telling us. In Northern Europe, the populations of E. punctata hibernate are inactive during the winter. So they are inactive during the winter, so there must be something to relate to with the temperature. As the temperature increases in early spring, March, the populations become active. The populations remain active and produce three generations before the next winter begins. So winter starts November, December, January, February. March they start to breed. Until November they have had three generations before the next winter begins. Wow, amazing. It was observed that in some areas in population of A. bipunctata, the proportion of the non-melanic to melanic phenotypes in the early spring was different. Because you see, they come out of this in spring. So early spring was different to the proportion later in the year in autumn. So March and autumn is then in October, September, October. So it was observed that some areas in populations of A. punctata, the proportion of non-melanic, the red ones, in the early spring was different to the proportion later in the year. So either the red ones were less or more, that we're going to figure out. Researchers carried out a study in one area of Germany to compare the proportion of non-melanic to melanic phenotypes in early spring with the proportion in autumn. First, they did this whole survey in spring, and then they compared it with the proportion in autumn, which is in September, after three generations had been produced. The data were collected over a period of 12 years. Wow, a lot of work, hard work, 12 years. The results are shown in figure 2.3. Now, This is the thing that we have to concentrate on. We have to look at the data very carefully and explain it to ourselves, the data. Now let's look at the data. We have the non-melanic and the melanic. Okay, let me shade it now. So the non-melanic, I'm going to color it yellow. So these are the non-melanic, the red ones. So that's all here. Then we have here, then we have here, then we have here, then we have here, and then we have here non-melanic. This is all non-melanic, right? A lot of non-melanic, non-melanic, non-melanic. Look at it carefully. This is all non-melanic. And the melanic, let's color it some other color, which is, this is melanic. So we have, this is the melanic, which is all this, then this is the melanic, and this is the melanic, melanic, melanic. So we can shade it like this. So I've just shaded it just for my own little comprehension. And what do we have on the y-axis? We have percentage frequency in sample populations of non-melanic and melanic phenotypes of the iBank data. So now if you look at it in autumn of 1929, what did we have? We have 70% of melanic. This is 70% of melanic. We have 70% of melanic. And we have about 30% of non-melanic. So this is 30% and this is 70%. Then in the spring again we have, now this is less, it's 30%. And we have 70% of the non-melanic. So you can look at it carefully, spend about 5 minutes going through it, write down all the things like I have done, that this is now 30 in this situation. Now this was in spring. All the springs, do they match? Then look at that. Do all the autumns match? So spring, and then in the autumn, it was in the later on, it was sampled percentage figure in sampled populations of non, and this was now 70%. So you see here, this was autumn, and now this is spring, and then autumn again. So compare all the autumns and the springs and see how you want to figure it out, because this was done over, so 19, 29, 30, 31, and this is to 1940. So it is 12 years, 12 years of sampling. So there are 24 samples. You see, this is 24 if you can count them. And then there was nothing this year, so this is also one. So you can start it from 1, 2, 3, 4, and 5, 6. This year there was no data, so this would be the 7th. So we have in 12 years, we have 24 samples. So this one would be 24th. Because in 12 years it was done twice, spring and the autumn. Then coming to part 1, it says outline a method that could be followed to collect the data shown in figure 2.3. Now, how do you count them? Number one, of course, the most basic rule of sampling is it has to be random sampling, or you can even do systematic sampling. So either you did random or systematic sampling, got your mark, and then you use quadrats, or you use sweep nets, or you use the Pooter. So any of these could have been used, a quadrat, a sweep net, and a Pooter. I'm just going to show you diagrams of that. And then you counted the number of each phenotypes. But even if you gave me the mark release recapture method, which is you catch them, you mark them, you release them, and then after a suitable time, you catch them again, and then of course you do the formula. You capture the second sample, and then in the second sample you count the number of marked ones in the second sample. But that could only get you 2 marks. Mark release recapture would only get you 2 marks, because that is not actually a very good method in this situation. So this is basically a pooter, and you can see here there is a plastic container, and then the place which you place over the animal, from then the plant, and then you suck up the animal. The insect falls into the jar, and then you look at the insect, and with the help of some sort of a book which classifies insects, you can then label them and call them whatever, and then you release them back into the wild. Now, this is basically a sweep net, and the sweep net can also be used to sample or to catch insects and then count the numbers and see how many were caught. And this is a quad rat. Then coming to part 2 of the question, the researchers concluded there was a change in the proportion of the two phenotypes between spring and autumn, with the non-melanic phenotypes always showing a higher percentage frequency in the spring. Fine. So the researchers concluded that the non-melanic phenotypes always showed a higher percentage. Now here comes the question. Suggest is, I always say, very difficult. Suggest two pieces of extra information about the investigations that researchers should provide to improve confidence in their conclusion. You see, if you look at the graph, now you look at the graph and see how things could have been improved. Now, first of all, there's no data here. Look at this. There's no data in the spring of 1929 and the spring of 1932. In the spring of 1929, so if they had provided us that, maybe that could have helped us a bit. Then the sample size, we didn't know what was the sample size. And then multiple sampling sites in same area of Germany. If they had told us where the sample sites were and where they were, and then they could have carried out a statistical analysis. That is the most important thing, because confidence improves when you do a statistical analysis. Now, they didn't tell us about any statistical analysis. Plus, they did not tell us something about was it the same location or the habitats or the sampling site? What was that? Where was that? Then was it done at the same time of the day? Or was it done some in the morning, some in the evening? What was the sampling time? And then the duration of the sampling. Did you do it over a period of 1 hour or 2 hours or 3 hours? They didn't tell us any information on that. You see, you must read the question very carefully. It says just two pieces of extra information about the investigation that researchers should provide to improve confidence in their conclusion. They concluded this, fine. That spring and autumn, that the proportion of the two phenotypes between spring and autumn with a non-melanic always showing higher in the spring. And then it says how researchers could could provide information to improve the confidence. So I'm just giving you some of these points, and that is data for the spring 29 and spring 32, then the sample size. We didn't know the sample size, then carry out statistical analysis. Of course, I've not given you all the points. You can look up the mark scheme for that. Now let's read the question. Environmental temperature is one of many different selection pressures that could be acting on populations of A data. The melanic phenotype, morph Q and S, are considered to be better adapted to the climate in northern Europe as they absorb heat energy more readily than the non-melanic type, the morph T. So the Q and S are better adapted because they absorb heat energy more readily than the morph T. Scientists investigated the effect of temperature on the body temperature of the two different phenotypes. The four individuals used in the investigation were collected while they were hibernating inactive and were kept outdoors for 6 weeks before the investigation. So we take out these four chungars who were inactive because it was winter, and we take them out and we leave them for 6 weeks before the investigation. So they have a party time for 6 weeks. And then the individuals were weighed and arranged into pairs, each with approximately the same mass. Then a temperature sensor was fixed to the lower surface of each individual. Poor things. The temperature of each individual was recorded for 9 minutes. During that time, a heat emitting lamp positioned directly above the individuals was switched on at 30 seconds, and switched off at 5 minutes. Poor things. I mean, they were exposed to 4.5 minutes of heat. The results are shown in figure 2.4. The results are shown in figure 2.4. Let's look at figure 2.4. Okay. So this is figure 2.4. You have figure 2.4 here. Please have a look at it. There is the temperature, and that tells you where is the heat lamp goes on, and there where the heat lamp goes off. Then the melanic phenotype, the non-melanic, and this is the air temperature. So let's first talk about the air temperature. So this is the air temperature. I'm going to mark it blue. So I've colored the air temperature in blue, and then we've got the melanic phenotype, which are the red ones. So I'm going to mark them here with the red ones. And then we have the non-melanic type, which I'm going to mark with a yellow marker. So the red ones and the yellow ones. So I mean, we are going to do this story with myself. Okay, so we've got the melanic and the non-melanic, and we see their temperatures. And this is the time when the light lamp is switched on, and so this is on, and then this is the time when it is switched off, and then we have the on and the off here as well. So this is the off, and this is the on. So at this time it was switched on, and that time it switched off. So look at this graph carefully. Spend at least 5 minutes on it. So melanic phenotype, which is the red one which I have marked, and the non-melanic phenotype is the yellow one which I marked. Then you can see heat lamp on, and then you can say heat lamp off. Now it says the researchers concluded that the melanic morphs will warm up more quickly than the non-melanic morphs. Explain how the information shown in figure 2.4 supports this conclusion. Wow. And then it says state two further conclusions that can be made from the results shown in figure 2.4. So first tell me, do they really warm up more quickly than the non-melanic ones? And then two further conclusions that can be made from the results shown in figure 2.4. And this is, by the way, only for 1 mark. And this two further conclusions is for 3 marks. So please be very careful what you write for 1 mark and what you write for 3 marks. Now when you look at this graph, it says Q and T males. Mass is 10.1 gram and 10.0 gram. So we have two of these here. And here Q and T males mass was 39.8 grams and 39.9 grams. Now where is the Q and T? Let's look at that. This was the Q, which is melanic, and T was the one which is non-melanic. So Q was the melanic and T was the non-melanic. So line for melanic phenotype is steeper than non-melanic. So line for melanic, which is the Q phenotype, is steeper than the non-melanic. Let's look at the graph. Yeah, you can see here Q and T. Q and T females. Melanic was the red one. So it is steeper. You can see in both of the graphs. You can see both of the graphs. It is steeper. So that is why I'm saying line for melanic phenotype is steeper than non-melanic. Then we come to part 2. State two further conclusions that can be made from the results shown in figure 2.4. Two further conclusions. Now we can make a lot of conclusions. Let's look at the graphs. All ladybirds have the same starting temperature. All ladybirds warm up when when lamp is on. All ladybirds return to original temperature. Melanic reaches higher temperature. Melanic cool downs faster. Females warm up quicker. This is the females. Females warm up quicker. Females reach higher temperatures. Females take longer to cool down. Heavier warm up quicker. You see, these were the heavier ones, 13.8 grams. These were 10 grams. So heavier warm up quicker, heavier ladybirds reach higher temperature, heavier ladybirds take longer to cool down. So you were going to compare the two graphs and you could come up with a whole lot of conclusions. All have the same starting temperature. So any three out of all these points, which I have mentioned earlier as well, and you can look in the mark scheme as well, all have the starting temperature, all ladybirds warm up when lamp is on, melanic reaches higher temperature, melanic cools down faster. So you could have come up, there are about, I think, 13 mark scheme points, and any three of these could have got you your marks. Now coming to the C part of the question, the scientists noted that there appeared to be a relationship between body mass and the increase in body temperature. The scientists therefore continued to take results from another 17 individuals. They recorded the maximum difference between body temperature and the air temperature for each individual in their investigation. All the results of this investigation are shown in table 2.1 and figure 2.5. So this is the table 2.1. Look at it very carefully. We have the T, Q, and the S, then we have their masses, and then we have the maximum temperature difference. Maximum temperature difference. So 0, 1.1, 2.7, 2.9, 4, 5.4, 5.9, 5.37. And then in the Q ones, we have 8. Well, you see what you have here is the mass in gram. So you can see as the mass is increasing in these also, the maximum temperature difference is also increasing. Then you look at Q and then you look at their masses when it's increasing. Well, this seems to be some sort of an anomalous result, but the others are increasing. And then the female seems to be having more. You see, this is a female 10.2. Then S, the masses are 8.6, 9.1, 10.8, and then we have 4.1 difference here. So as the mass increases, the difference also increases. Look at it carefully and sort of explain it to yourself. Then it says maximum temperature difference here, and then mass here, and says state. No, I think there's something before that. No, there's nothing before that. State a statistical test that could be used to analyze the relationship shown. Relationship shown in table 2.1 and figure 2.5. Justify your choice. Now, what could you have? It's a very simple clue they're giving you. Relationship. So Pearson's linear correlation and Spearman's rank correlation. You could have said either of those. So data is paired, data is continuous, scatter diagram shows a linear correlation and at least 5 paired observations. Then coming to the D part of the question, the researchers assumed that the prevalence of the elytra was controlled by a single gene with two alleles. Two alleles mean big A and small a. Breeding experiments showed that the melanic phenotype is dominant to the non-melanic. Researchers made the hypothesis some melanics, it says melanic is dominant. Some melanics are homozygous dominant and some melanics are heterozygous. Outline a breeding experiment that would be carried out to test this hypothesis and state the results you would expect if the hypothesis is supported. Cross the melanic with the non-melanic. Why? The test cross. If melanics are dominant, then you cross them with a homozygous recessive. You see, if you cross this with a homozygous recessive, then all the offsprings will be this. And if you have it's a heterozygous and you cross it with a homozygous recessive, well, some will be this and some will be this. So these are the two things that we talk about very often in a test cross. So you cross the melanic with the non-melanic, homozygous dominant will only have melanic offsprings, and homozygous heterozygous will have both melanic and non-melanic, because they've said melanic is dominant. So the dominant can only be this. Dominant can only be two genotypes. phenotype will be 1. Then you can give me details about the breeding experiment, many breeding pairs, each breeding pair kept separate from the other pairs, and count the different phenotypes of offspring. So mark scheme point 1, cross the melanic with non-melanic. Then mark scheme point 2, you tell me about the homozygous dominant with homozygous recessive and heterozygous with. So this was the point that you would give me. I've given it to you in a sort of a genetic diagram. Then many breeding pairs, then each breeding pair kept separate, and then you count the phenotype of the offsprings. Now you can read the exam report, and that teaches you a lot of things. A number of candidates. Now you can see here how mistake was done. A number of candidates use the term species when describing what they would count rather than phenotype. So instead of using the word phenotype, you were using the word species. All ladybirds were of the same species, therefore this did not gain any credit. Then part 1, successful answer to the fact that the line on the graph was steeper for the melanic phenotype compared to the non-melanic. Then the outline the breeding experiment that could be carried out to test the hypothesis. Some melanics are homozygous, some are heterozygous. They would expect if the hypothesis was supported. And then of course you gave me that. So please everybody read this. Please pause the page here, pause the video here, and give it a good read. Thank you very much.